everybody. I'd like to just start by apologising for the slight delay and to thank you all for joining today's webinar where we will be discussing antibody and testing validation. Before we begin, we just have a few housekeeping rules. We would appreciate it if everybody could keep all of their phone lines muted. There will be pauses for questions and answers during the presentation, and questions can also be submitted by the question text box. The presentation will be recorded and may be used later on Horizon's website. However, no Q&As will be posted. Since we are a public listed company, we're obliged to show you this disclaimer. I would now like to introduce our two speakers. The first is Dr. Regan Fulton, the Director of Research at Phenopath Laboratories, USA, and the second is Dr. Farah Patel, the Senior Product Manager leading development of IHC reference standards at Horizon Discovery. I will now hand over to Dr. Fulton to get started.
Good morning. Good morning. I'm coming to you from Phenopath Laboratories in Seattle, Washington. This is a collaboration. Validation. There's a slight delay, and I apologize. I'll get used to that delay. First, I want to take a moment to mention Phenopath. This is the reference laboratory in Seattle, Washington, founded by Alan Gown, providing a broad array of consultation and technical services to the anatomic pathology community. And because of your attending this conference, I thought you all would be interested in knowing about a newly created society called the International Society for Immunohistochemistry and Molecular Morphology. We have a website, isimm.org, and we are just getting started, but from this group you can expect discussions and educational programs concerning quality control and advancing knowledge in the area of slide-based testing. So very relevant to today's talk. The outline of today's talk, we'll speak briefly about optimization, verification, and validation of slide-based assays, and then the importance of standardized reference material for these processes, and speak to the usefulness of on-slide controls, particularly in the context of PD-L1 and HER2 for the purposes of this talk. So let's discuss the distinction between optimization, verification, and validation. Optimization is the process by which an individual laboratory maximizes the sensitivity and specificity of an assay. And it's reasonable to stop here and talk about what the implications are of a failure to optimize an assay. Now, this comes before the validation process, but it is very much a part of it. Our friends at the CIQC, the Canadian Immunohistochemistry Quality Control group, including John Garrett and Emina Torlakovich and Blake Jilks and others, Carol Cheung, all of whom are members of ISIMM. They looked at surveys that they distributed in a TMA format to about 80 laboratories, and what was interrogated was the lab's ability to detect some fairly simple normal structures as well as poorly differentiated adenocarcinoma with either a pan-keratin reagent or a low molecular weight cytokeratin. And as you can see in these two lower rows, Laboratory A and Laboratory B had not fully optimized their reagents, so that they were not, with these assays, the pan-keratin or the low molecular weight cytokeratin, able to detect hepatocytes, kidney tubules, or in one case, poorly differentiated adenocarcinoma. They were not able to define the epithelial nature of this tumor. Admittedly, there's some low level of staining in the lower right-hand corner. However, wouldn't necessarily consider that firm evidence of epithelial differentiation. So you can see that this is a cautionary tale regarding the optimization of an assay. So with respect to method optimization, there are a number of parameters that need to be considered, including tissue ischemic time, fixation buffer, and plus or minus decalcification if appropriate, the fixation time and the processing method, among others. And increasingly, this is being recognized as part of the establishment of the performance characteristics of a particular assay. And our friends at Roche-Ventana, in the release of their SP142 assay for PD-L1, have demonstrated a fairly robust examination of the pre-analytic conditions and their effect on PD-L1 staining. So you can see across the top a variety of different fixation buffers, including neutral buffer formalin, zinc formalin, and others, and different fixation times down the left-hand side. Optimal staining is seen with neutral buffered formalin with 6 or greater hours of fixation. So with this kind of analysis, you can clearly see that pre-analytic factors play into the performance of an assay. And I think more than the specifics of this particular assay, the importance of this kind of analysis and the release of it with the documentation associated with a new assay is kind of a sea change for us in the immunohistochemistry world. It reflects a greater need to clearly define the parameters by which assays are run, and I think we'll be seeing a good deal more of this, both on the part of vendors when they are releasing new assays and end users who are going to have to look at their own conditions prior to running their own assays. So in optimization, we want to look at pre-analytic factors, and then there are analytic parameters, including the selection of a staining instrument, the antibody itself, the retrieval method, the titer and detection methods, among others. I'll just speak briefly about the retrieval method and show some dramatic images of the importance of the appropriate retrieval. Here at Phenopath, we use a variety of different conditions to retrieve antigen, and you'll see that there are really significant differences in different assays. So in our hands, we have very broad, a broad array of antigen retrieval conditions. I realize that some people on automated instruments may have less flexibility in that regard. But here is an example of TTF1 with citrate retrieval pH 6, which we consider optimal. A higher pH, the EDTA, shows much less nuclear staining, and still less with tris buffer at pH 10. So you can see that the sensitivity of the assay goes down considerably as these buffers increase in their pH. So that is definitely something to be considered when optimizing an assay. And so before bringing an assay on board, the pre-analytical parameters, the analytical parameters, and appropriate control material need to be considered. And with respect to that control material, we can think back to the CIQC survey where liver and kidney and poorly differentiated adenocarcinoma were not detected. One can speculate that those laboratories had not used those appropriate targets during optimization procedures. A brief mention of verification. This is an abbreviated validation process for FDA-cleared or approved reagents, and it's applicable also to certain laboratory developmental tests that changes therein, including new antibody lot or change in heat retrieval conditions, etc. And we'll briefly return to this later, but just understand that this is a very truncated form of validation after receiving a FDA-cleared kit or changing a pre-existing assay in minor ways. So IHC validation. Why do we do it? Well, one of the big reasons that's articulated by the CLIA requirements are that we must establish and verify the method performance of any particular assay with respect to accuracy, precision, sensitivity, and specificity. And we'll give some further discussion of those aspects. So there are two categories of validation, and we'll focus, of course, on analytical validation in this talk. These are the procedures to confirm the ability of an antibody to recognize its target in normal and diseased tissues. So that's the analytical component. The clinical validation are the studies to confirm that the assay predicts clinical outcome. Now that's generally well beyond the scope of any particular end user. So we will clearly be focusing on analytical validation in our day-to-day practice. So as part of that, we are tasked with assessing the sensitivity, which is to assess whether the assay detects the target analyte when the target is present, and the specificity, which is to make sure that the assay does not detect the target when it is absent. So once again, returning to this very instructive survey by CIQC, the sensitivity of these assays was quite insufficient. And it's important to clearly elucidate that along the way. If it hasn't been done at the optimization phase, then the validation phase should catch it and re-optimization would be indicated. Now with respect to antibody specificity, again, pre-analytical variables, analytical variables, and tissues selected as true negatives are important considerations. This, for example, is an image of PD-L1 stain in tonsil and was published on a very well-known website as an example image of PD-L1 staining in tonsil. And as you can see, it's just a wash of brown. There's no specificity whatsoever. You can't tell positives from negatives. So it's important to know what the expected reactivity pattern should be. And when you don't see the expected pattern, that is, that positives are positive and negatives are negatives, to go back, re-optimize, and then revalidate. The other CLIA requirements are for accuracy and precision, and these are under the broad rubric of reproducibility. So they're not exactly the same. Accuracy has to do with how close to the right answer one is getting, and precision has to do with how repeatable your result is. What we strive for is high accuracy and high precision. And these are generally measured through reproducibility studies, primarily through intra-technician studies where positive controls are run in multiples, preferably triplicate or greater. Inter-technician, where a second technician will repeat that experiment. And then inter-day and inter-instrument experiments are also very helpful to establish reproducibility. Standard validation methods are a little bit variable, but in general they revolve around the following concepts. Expected staining pattern and distribution of tissues in a mix of normal and neoplastic tissues is what we strive for. Now, what defines the expected staining pattern? It may be knowledge of expected reactivity from reading the literature, or having tissues that have been previously characterized either in one's own lab using a different assay or in another laboratory using either the same assay or a different one. So those would be gold standards for the purposes of a particular validation method. That is, the expected staining pattern would represent your referent, and your new test would be compared against that. Now, this just repeats that concept. The identical pattern of staining with different antibodies directed against different epitopes would be an inter-method comparison, broadly speaking. Now, there are more advanced validation methods. We perform these routinely at Phenopath, as do many other laboratories, and we look at the correspondence between IHC and other methods such as DNA or even RNA in situ hybridization. RNA in situ hybridization is a newer technique that is provided by ACD in the form of RNA scope as well as Affymetrix with another proprietary assay. So that correspondence can be very helpful. That is another type of intermethod comparison and can inspire great confidence in specificity in particular. And another more sophisticated method is looking for alterations in the staining in genetically modified tissues following either transfection, siRNA exposure, or even modification at the genomic level. And we'll talk a little bit more about such tissues later in this talk. Other advanced methods, less used in the routine workup, include looking at Western blots with the assay, the antibody of interest, and then disappearance of immunostaining after pre-absorption of the antibody with a purified epitope. So a competitive assay. Now, how about real-world validation? No talk would be complete without some mention of the new CAP recommendations. These were published in 2014 as an attempt on the part of the CAP to provide some guidance as to what should be the minimal validation and revalidation requirements for CAP and CLIA certified laboratories. This was a recommendation of a panel of pathologists and histotechnologists with expertise in IHC who reviewed the existing literature and addressed a variety of different issues based on existing evidence. Now, it turns out that the evidence was not sufficient in many cases to make firm recommendations based on the evidence, but rather much of this was expert opinion and consensus of the committee. But the recommendations are summarized as follows: that all tests must be validated prior to introduction as clinical assays. Concordance in validation studies was suggested to be a minimum of 90%. Now, that's of course variable and is at the discretion of the lab director, depending on the availability of tissues and the robustness of the assay. So for non-predictive assays, the minimum number of positives and negatives to be included in a validation study was 10 and 10, respectively. But for predictive assays, such as ER, PR, HER2, and others, 20 positives and 20 negatives were considered the minimum. And if any particular assay, such as ER, was used in both contexts, then the higher threshold should be used, that being 20 and 20. Now, it was recognized that pre-analytical variables could play into the results of the assay, and so it was stated that when possible, one should use validation tissues processed with the same fixative and processing methods as clinical cases. Now, it's important to state that that doesn't mean that the tissues need to come from one's own laboratory, because in many cases it's clearly not possible to obtain those tissues internally. So that is an attempt to clear up some misconceptions about the statements regarding using internal tissues. It also reflects the advancing nature of our field, where more and more obscure diagnoses are being made with limited tissues that have been previously characterized in that respect. The use of cytologic material, again, recognizing the pre-analytic component affecting the assay. The suggestion is that the laboratory director should determine a sufficient number of positives and negatives to analyze in that respect. Similarly, the decalcified material should be looked at with a sufficient number of cases at the laboratory director's discretion. Number 9 was that TMAs are okay. Large whole tissue sections were considered optimal, but TMAs showed sufficient concordance with whole sections as to be appropriate in the committee's opinion for validation purposes. When a new antibody lot was introduced or is introduced into clinical use, at least one positive and one negative should be run to confirm the reactivity pattern is equivalent to the prior assay lot. And the last 4 recommendations are shown on this slide, that when an antibody dilution is changed, the vendor of a particular clone is changed, incubation time, and other such minor tweaks, that there should be 2 positives and 2 negatives run to confirm equivalence. But when perhaps more important or more dramatic changes occur in the assay, such as fixative type, retrieval method, detection system, etc., then sufficient numbers of tests should be run, again at the discretion of the laboratory director. If a new antibody clone is introduced, that's essentially introducing a new assay in the opinion of the committee, and therefore 10 or in some cases 20 positives and negatives should be run. And finally, the archiving of documentation should be kept for a minimum of 2 years. So let's look at an example real-world validation. In this case, we're looking at 20 positives by some gold standard and 20 negatives by the gold standard. And again, defining a gold standard can be quite difficult. We may be talking about expectations of reactivity within normal tissue based on the literature, or intermethod comparisons with other antibody assays, or interlaboratory comparisons with the same assay. In any case, we'll take, for the purposes of this talk, for granted that the positive cases are actually positive, negative cases are actually negative, and we'll call those the gold standard. Now, in this example validation, those same 20 positives and 20 negatives were subjected to a new test, and there wasn't a 100% concordance. You can see that 5 of the gold standard positives ended up in the negative category in the new test, and those would be the false negatives, and we had 2 false positives as well. So an example calculation, I like to use a little Excel spreadsheet calculator for quick and easy determination. Plug in the values, and we get a positive agreement or sensitivity of 75%. That follows directly up from 15 out of 20, so 75%, and a negative agreement or a specificity of 90%, 18 correct negatives out of a 20 total cases run. So 75% sensitivity and 90% specificity is what was achieved in this example. It would be at the discretion of the laboratory director whether to accept or reject this validation result, although the CAP recommendations are for a 90% concordance. Now, we should distinguish concordance on the positive and negative side, those specifics from overall concordance, which is essentially the average. And overall concordance in this case was 83%. That calculation, of course, is taking the total number of concordant results, both positive and negative, and dividing by the total number of cases. So let's look at one of these more challenging situations where there's very limited tissue availability in a laboratory for a particular new assay. Let's say NUT1, for example, for nut midline carcinoma. ALK in a particular laboratory may not have previously characterized tissue, and those tissues may be hard to find. Many other examples exist. So what happens when a laboratory is faced with an N of 1 for gold standard positive? And of course, in collecting tissues, there's any number of negatives that one could run, and if one stacks the deck and uses 50 negatives, what kind of results can we expect? Well, let's say in this thought experiment, the new test shows a positive concordance, 1 of 1. There are 2 false positives by the new test. They were gold standard negative. What kind of results do we get there? Well, the positive agreement, of course, is 100%, and so we would report the sensitivity as 100%. The negative agreement was 96% because we had so many gold standard negatives, and the overall concordance would then be 96%. But can these numbers be trusted? Well, in the context of validation and statistically, we cannot really draw too many conclusions from that. It's we're on thin ice to use these numbers confidently, and this is the kind of consideration that motivated the committee to set a minimum number of cases, yet there is that recognition that cases can be hard to find. So where do we find appropriate validation samples? Well, the first place to look is your clinical case archives, if available. Another way of obtaining tissues and validation samples is to do interlaboratory sharing, and one organized method for that is a fledgling sample exchange registry from the CAP. That's something I would encourage people to look into. It's essentially crowdsourcing of tissues, which is a brilliant idea in so many different contexts, and certainly could be done with tissue validation. CAP and other external quality assurance agencies provide proficiency testing surveys. This can provide excellent material and interlaboratory concordance data for doing validations. And finally, and importantly for the remainder of this talk, is the commercial sourcing of both tissues and cell culture. So, standardized reference material. In this era of companion diagnostic assays, which are increasingly quantitative, it's difficult for us to calibrate our assays based on remnant diagnostic material for all of the reasons that have previously been stated. The pre-analytic variables, the lack of experience with a particular assay, and the variabilities therein. So standardized reference material strives to control for ischemic time, fixation time, processing conditions, and tumor heterogeneity. And these materials would then, under ideal circumstances, be available for optimization, verification, validation, as well as daily controls. So the assay that we referred to previously, the SP142 from Roche Ventana, does not come with its own internal control, but rather they suggest using a tonsil, and they provide images of the expected result. And so a laboratory is obligated then to obtain tonsil and assure through pre-qualification that it represents a good control, and essentially it becomes wallpaper matching to make sure that the results obtained with your tonsil tissue match those shown in the data sheets or product inserts. But what if we had precisely defined, genetically engineered cell line material that was previously rigorously analyzed by a variety of different methods that we could use on a daily basis to show different levels of expression of the markers of interest. And in this case, I'm showing examples of PD-L1 staining. Now, these are in fact horizon cell lines, and they show just what I've been speaking of, this genetically engineered and rigorously defined expression levels, which could be so useful in the context of reproducibility studies, daily quality control, validation generally. So in PhenoPath experience, we've looked at these cell lines and done a variety of experiments, and these have helped us with our validations and verifications of different assays, including the FDA-approved 28-8 assay, the 22C3 assay, LDT, and even the RNA-scope assay for PD-L1 that we worked up as an LDT. So you can see the usefulness in terms of determining reproducibility. In this set of runs, we can see slightly lighter staining on the third run for the 28-8, whereas there's very good consistency here in the 22C3. This is not necessarily representative of one's overall experience, but again, this illustrates the potential value of these kinds of materials. As I said, we have worked up the RNA scope, and it provides an important inter-method comparison, which can bolster one's confidence in specificity as well as sensitivity of a particular assay. So that's on TMA whole tissues, and the same is shown here using cell culture material. Horizon also provides HER2 cell line control material, which is again very rigorously defined and provides a very nice spectrum of 0, 1, 2, and 3+. Now I hear a voice in the back of my head coming from 15 or so years ago when I was starting my career, and essentially crying out for such material to be used on every patient slide so that one could calibrate the assay in the context of a given slide and really know what the assay conditions were on that particular slide so one could make a confident diagnosis of HER2 expression in a particular patient. And this has essentially become a possibility based on the availability of such material. Here's an example of a HER2 reproducibility study that we performed here at PhenoPath, and we see very good reproducibility across these 5 runs with the 0s, the 1s, the 2s, and the 3+ cell lines. And now a mention about that mantra of mine that while internal controls are best, these on-slide controls are at least substantially better than batch controls. And in fact, CAP has long said that the best practice is for multi-tissue on-slide controls to be present on patient slides, and they're a better indicator of the test working on a given slide. And these can, in fact, be cost-saving in terms of labor and materials, as well as space on instruments. And it's an important consideration that in today's random access instrumentation, where batch runs can be considered as small as 10 slides per tray, for example, that to have batch controls for each different stain would really take up an inordinate amount of space. So for example, if you're running 5 markers on a given case, if you had to run 5 separate controls, that would take up the whole tray with 5 and a 5. If instead you can consolidate onto one slide with on-slide controls, you're really saving instruments space, and that can have all sorts of downstream implications. So here is an example of running PD-L1 cell line arrays on the same slide as target tissue. And one can see that the assay worked and had the appropriate level of sensitivity and specificity very quickly and easily. Similarly, we can do this with HER2 and a variety of other markers. Now, the cell lines are also useful in the world of in situ hybridization. So these cell lines are interrogated with HER2, and we have both negatives and clear positives that correlate nicely with the expression levels. So to summarize, all assays should be optimized, verified if appropriate, or validated, and need daily quality control. This would require appropriate reference material, and while internal material can be appropriate, it is often difficult to qualify appropriately, and so that those remnant tissues, our third point here, they are difficult to obtain and qualify. We need to control for pre-analytical and analytical variables. In terms of the daily QC, same slide controls are clearly best for a variety of reasons. Better still, of course, are internal controls within the same tissue, but they're not always present. And finally, genetically engineered and extensively characterized control material is now available, and I would encourage end users to consider using it where appropriate. So with that, I'll hand this back to Farah for her few words as well. Thank you.
Thank you, Regan, for the excellent introduction and insight into the world of antibody validation and highlighting the importance. So I'm Farah Patel, and the Senior Product Manager leading the IHC Reference Standards Development Program at Horizon, based out of Cambridge in the UK. As we all know, immunohistochemistry has evolved from an investigative tool into a widely used diagnostic technique within clinical laboratories. It serves as a diagnostic and prognostic assay, and in clinical settings, it increasingly contributes to the choice of treatment. And more so with today, IHC has become is in the spotlight due to the development on PD-L1. Although it has been employed for 50 years, the application of this method suffers from lack of standardization and reproducibility among the different laboratories. So today I'm going to touch upon variability in the IHC workflow, but most importantly, how do we develop these defined genetic reference standards as independent quality material for everyday use to address the inconsistencies and issues around reproducibility and consistency of your assay. I would like to highlight at the beginning of this presentation that these reference standards are for research use only. For the next 10 minutes, I will also discuss PD-L1 and HER2 reference standards with specific importance on its development and how to use it as a tool for optimization and verification activities. So before I start, a very quick summary on who we are. Horizon Discovery is a cell building company. We are a company based out of Cambridge, and we look at all aspects of cell function. We drive to understand the interplay between DNA, RNA, and protein, and how these aspects impact the laboratory workflow. So Horizon is building these human disease models and reagents from genetically engineered cells that our customers use or that we deploy on their behalf, and that is to gain knowledge on genetic drivers of disease, develop novel drugs, or cell therapies. We are increasingly passionate about developing and delivering high-quality, genetically defined standards as controls for these different assays. So from this understanding, of the genome and the protein, RNA, and DNA, we've developed a gene editing technology, and using that technology, we can manufacture standards that are precisely defined. For example, for the genomic DNA, we know we can go all the way down to the different allelic frequencies that will test the sensitivity of these assays, and then you have the FISH and IHC, and the cell-free and RNA standards as well. But today I will focus more on the reference standards on the IHC side of things. Now, I'm sorry, I do apologize earlier. We started a bit late because we had a few problems with our slides, with my slides in particular. So we had, so a bit of the introduction has been, 3 slides of my introduction have been erased. So what I would like to do is give you a quick summary of what I was going to say about the IHC workflow and how the variability can creep into the standard IHC workflow. We all know that there's fixation, there's sample preparation, there's antigen retrieval, antibody staining, the amplification step, and then you finally have the microscopy. And now more than ever, you also have QDP, which is quantitative digital pathology, to quantitate the intensity of your staining per cell. So all these different steps in a standard IHC workflow can lead to variability and errors, and it's not sometimes nobody's fault. It's just that it is the way it is. IHC has too many different components, and it's very hard to have a standard assay for routine use. So most of this is covered in our previous Horizon CAP webinar, and that's on our website, and you can go into that in more detail. But the most important thing I wanted to say is understanding each step and identifying where variability can creep into the workflow is very important. And the other point I wanted to mention very quickly is, understanding these workflows has a big impact on the clinical pipeline. More so again now than ever, where IHC is used to treat patients directly for targeted therapy. Another good example is PD-L1. You have the importance of having a reliable, consistent, and accurate assay is crucial for all these stages, whether it's from R&D in the assay development stage, all the way through clinical trials, and all the way through to the customer, to the end user at the end of the day. So good assay validation leads to better trial design and improves chances of patient selection, market success, and I think most importantly, delivering the right diagnosis and prognosis to the patient. So this brings us to the rigor and why we do what we do at Horizon is to build tools for assay optimization and verification for IHC. So what we did was our objective, as I said, was to answer some of these questions and these challenges by developing this tool set, which is negative and positive on-slide controls with low, medium, high expression that could be used as an on-slide control for routine use. So the fundamental question I think you must ask yourself, and all of us ask every day, is, has my IHC assay worked? Is my IHC assay optimized? But most importantly, has it worked? So using Horizon's precision gene editing technology, what we did was we wanted to create a quantitative, precisely defined PD-L1 reference standard. So we used a new technology where we engineered PD-L1 using a landing pad technology where different clones were generated. These engineered clones were fixed, pelleted, and processed into a 15-core development array, as you can see here. And that was tested internally and externally. In addition, each cell line you can see in this core were extensively characterized by sequencing, gene expression analysis, and digital PCR. Here you can see a beautiful range, a dynamic range of protein expression. You have the high expressors on the left-hand side, and you have this lovely range of low and intermediate expressors that would test your sensitivity of your assay. And then you finally have the negative control, which is your wild-type control, which always should be negative for PD-L1 expression because it has no PD-L1 in it. So that is always your benchmark. What I also wanted to add here was the 15-core development array that was developed with this range of expression cell lines. 4 of them were selected and used in our final product in number D, as you can see here in the FFP blocks and FFP slides. What we've done also is we've extensively used QDP to analyze and assess this 8 score, that is the intensity of staining, but also the percentage positivity in the cell lines. So you have the negative range, as you can see here. The negative cell lines are all the way down, have a very low 8 score, and then you can see the beautiful dynamic range going up. And then you can see the top high expressors with an 8 score of over 200. So once we have these cell lines, then we can, as I said earlier, it goes through a different series of quality control. The blocks are manufactured, these homogeneous cell blocks are manufactured, and then they're cut and mounted on slides. So building reference standards is a big responsibility, and hence we have stringent QC criteria at Horizon. This slide demonstrates the following. The first is the dynamic range of PD-L1 protein expression was detected in the cell line microarray. Here, we've used the cell signaling antibody and the SP142 antibody from Ventana, but we've developed our own assay, an equivalent to an LDT, to see the dynamic expression. What we also see here is, if I'm just going to go by step, you see core 1 with a negative, core 2 with a low, and core 3 with an intermediate, and then core 4 with a strong. And you see this beautiful reproducibility between both antibodies. Figure C shows you the consistency in protein expression between 2 independent cell groups. Most people have a very negative impression on cell lines because they never get enough consistency and reproducibility. Well, we have in our manufacturing process, we have our cell lines. We know what we need to do to keep this consistency alive. And one of the things we always do is measure the consistency of the cell line by quantitative digital pathology. So batch-to-batch reproducibility in the cell growth and manufacturing process is tested here. Figure D shows you that the PD-L1 IHC reference standards are compatible with RNAscope. Figure F. In this image, ACD has used a probe 8CD274 and the RNAscope 2.5 LS kit, and that was run on the Leica RX. And as you can see, a very clear increase in mRNA expression is seen across the low, medium, and high protein expressing cell lines. And finally, we have the RT-qPCR analysis using RNA extracted from individual cell lines from these FFPE cores that show consistency and consistent results with the RNAscope, but as well as the IHC data. So the next step here was to, once we had an idea of what we were doing with our array, we wanted to send it to the manufacturers and also to top laboratories who would use our array as per the manufacturer's standards. And you can see here the 4 assays have been tested. You have the two 22C3 and 28A. You see a very clear dynamic range. The negative, low, medium, and high are expressed and are concordant with on-site tissue controls. The SP263, you see a very clear dynamic range again as well in the 263 antibody using the manufacturer's controls. But in the SP142, you lose that dynamic range. You always pick up the negative and the strong, but you do not pick up the low expressors. And again, let me please remind you that this is as per the manufacturer's instructions. So the next step here was to send this to some external labs. And I've just picked two very small examples to show you out of the many labs that when we sent it to these labs that had validated protocols or were in the process of optimizing their protocols, I think 3 out of 10 to 15 labs got this right. Most of them were trying to optimize 3 to 4 different protocols using these different assays, and they had mixed results. Most could detect the high expressor because you've got the high protein expression in the cell line core. And here you can see a laboratory that has used a laboratory developed test does not pick up the medium and low using the SP142 antibody. Now we have shown, and many others have shown, that the SP142 antibody can indeed pick up PD-L1 protein expression, but here you do not see that. So there's loss of sensitivity, so there would be a possibility of having a false negative. Again, this is another example where you see false positive, and this is using the DACO antibody. Here we have seen, just one sec, yeah. We've seen here a very nice range of protein expression, but here this laboratory has a false sort of a background staining, a false sort of a blush, false positive staining in the negative core, and when we've measured this by QDP, you see a drop in intensity. So coming back to HER2 now, what we did is we had a lot of success with the PD-L1 and this new technology that you could quantify the protein expression. We used that to engineer the HER2 standard, and again you have a very lovely range of negative 1+, 2+, and 3+, and we always measure the intensity score and the percentage positivity using quantitative digital pathology by Visiopharm. We work in partnership with them, and we've developed these biomarker-specific algorithms that detect and can help us analyze the intensity of staining. So, just another quick thing I forgot to mention here was, when we talk about antibody validation, there's more and more need of having these cell lines that do not actually have the protein in it. So what we are doing now is we're actually knocking out the protein of interest. In this case, we've knocked out HER2. So it's a complete knockout, so there should never be any staining, any positive staining in the cell line. And if you did see a positive, observed a positive staining in this cell line, it would suggest that there is a problem with the assay, and one would suggest that you repeat it. So this is your true negative, and this gives you the confidence, whether it's when you're developing an assay or optimizing it or setting it up, that this is your benchmark. It's your true negative. And similarly with the ER, we've again created an ER gene knockout, which is quite tricky because it's a massive gene. We have, so again, you should see no protein expression here, and you have this lovely range of intermediate and strong in your ER. So to conclude, I just wanted to run through a couple of key features and applications. Most of you now would have understood that these defined cell line cores are important, can be used as on-slide controls, and it gives you confidence through verification and validation activities. They test the technical performance of your assay, and they help you establish the practical lower limit of detection of your IHC methodology. And most importantly, they guide the evaluation of antibodies based on their sensitivity and specificity. And again, these reference standards are for research use only. If you need any more information, please do not hesitate to contact me or us at Horizon. And a question we always want you to think about is, what is the impact of assay failure in your lab and how do you monitor for it? Thank you. We're just going to give us 2 minutes and we will just check if any questions have come in, and we will be with you shortly. 2 minutes. Yes. So we've got a question here from one of our participants. A question for Dr. Fulton. Dr. Fulton, are there any, what are your thoughts on the PD-L1 antibody validation currently?
You've shown there can be quite a variety or a variation in reactivity patterns as compared with reference standards and other assays on those reference standards. So I think it's difficult with respect to the complementary diagnostics that are coming out that are FDA-approved kits. The validation is somewhat truncated in the form of a verification, and I think that's relieves the end user to a certain extent of the burden of doing high-level validation, but the daily quality control, I think, is still tremendously important, because with at least one of those FDA-approved assays, we do see quite a bit of variability, and particularly high background. And so on those occasions, I think it's important to consider repeating the test, and it's hard to really know that the test is not performing without a good control.
Regarding inter-instrument reproducibility during the validation process, how many would you suggest using? Would you repeat all the 20 samples per instrument?
I think that's next. I think it's next. With 3 control slides known to harbor just 3 sections of a single well-qualified control, and make sure that those 3 slides match 3 slides performed on another instrument at another time. And one can also look at inter-technician reproducibility and inter-day reproducibility with the same kinds of numbers of slides.
Tissue and positive control tissue on each IHC slide, or just a known positive control. How do we guard against false positive results on patient cases?
CAP has already made the recommendation that reagent controls are no longer necessary in the age of polymer detection, so that background issues have effectively been eliminated in most circumstances. But tissue controls are another story. We really have to look for those internal negative elements primarily, but they are not always present. For example, in samples that simply show tumor. And so having a control that contains both positive and negative elements is tremendously important. And once again, my strong opinion in this area is that on-slide controls are best.
To you, I think our marketing team will be able to send you all the participants an email with the link. please do not hesitate to contact us. And thank you very much, Regan, for doing this webinar with us. And I hope everybody found it extremely useful. Thank you very much.
Thank you, Farah. And I will mention for references on some of these issues. And what's not included, because it's still in preparation, is a series of opinion papers coming out of the International Society of Immunohistochemistry and Molecular Morphology. These papers are headed by Amina Torlakovic, and I think will be a very important contribution to this area. So thank you very much for allowing me to participate.